So let us welcome Daniel. Please.
Thank you. Thank you. So I'll try my best to keep it as short as possible. I know everyone is looking for the coffee. So I'll give you a quick presentation of what holotomography is and how you do it and how Tomocube is doing it at the moment. It was really nice to see so many really cool applications this morning. I missed a few of the talks yesterday, but also seeing some new techniques that other people are using. And I just want to show you another technique that would give you some insights into your research. So I want to show you just this video here on the left. So if I were to ask you to guess what the colors are, probably one guess would be, oh, those are 3 different fluorophores that I'm using, like blue, yellow, and red, and so on. In fact, what you see in there is refractive index measurement thresholding. So basically, a refractive index measurements in 3D, where you say from this number to this number is the blue, from that number is the yellow, and so on. So what happens here is live imaging in 3D, where a T cell is attacking a cancer cell. And you see the interaction between the 2 cells in 3D. And upon that interaction, you'll see that those really dense objects inside the cell starts increasing within seconds. So you can do this over time. In reality, the data looks like this. So it's gray levels. So we measure refractive index in 3D. So whenever you see this word, holotomography, think about 2 words, holography and tomography, the fact that it's in 3D. So normally whenever I give this presentation, my main joke, or whatever you want to call it, is whenever we talk about holotomography, I want you to remember 3 things. Holotomography is 3D, is always 3D, is quantitative, but it's also label free. So those are the 3 words that I want you to remember from this talk, if nothing else. And of course the pretty pictures will, which I'll show you in a second. But in doing so, now you can quantify individual things. So you can segment in 3D the objects of interest. So we're looking inside the cells. We're not looking at population of cells. We're looking inside of individual organelles of a cell due to the resolution and the measurements themselves. So, as you probably know, I'm not going to go through all the steps, but what we're trying to do here is achieve imaging in 3D live. And as you know, in many techniques out there, you have some limitations or difficulties to get your cells happy or keep them happy or alive due to the phototoxicity and so on. And in many cases, it takes a longer time and it's usually qualitative, not necessarily quantitative. So I'll give you some principles, but I'm not going to go into the physics of it. So sorry for people that really want to go into the depths of it. I'm not a physicist, but I'll try my best to explain what we're doing here. But before we do that, in general, what we're trying to do here is to be able to image in 3D really precious samples. I know in many cases we take the cells, we fix them, and then we image them, but you cannot go backwards in that case anymore. So what we're trying to do here is to solve this problem. We call it pyramidal frustration. So as you know, in many cases you need to play with the signal to noise ratio, or you have to keep the cells alive, or is the temporal resolution, and so on. So what we're trying to do here is to minimize that pyramidal frustration with the new method that we launched back in 2022. And that, for some of you that do not know what the holotomography is, I want you to just think about 2 other techniques out there that are quite known, so bright field and fluorescence microscopy. What we're trying to do here is bridge or find the best parts of other techniques and keep them, or even get them more robust in some way. So as you know, in fluorescence microscopy, definitely in fluorophores. In holotomography, you don't need to. But the main thing here in holotomography is we're trying to image really fast. So that means your cells are not, even though they are moving potentially, you can quantify them. So you don't have to, we freeze them in time basically when we image. So that's why it's in seconds as compared to minutes, or depending on the techniques out there. And it's also 3D. And due to the resolution, I know some of you are going to ask me, why is it below 200? And that's the magic of it, which I'll mention in a second. And it's always quantitative. So that means whatever pixel you see here, that's a voxel. So in each voxel, you have refractive index measurements, which means you can quantify the dryness of those lipids in those lipid droplets of those proteins inside the mitochondria and so on. How you do that? Well, the principle behind the holotomography is under the same umbrella as normal QPIs, I'll say, but it's in 3D. In our case, in general, you have the wave front, so you have the light passing through the object, the light scatters, you measure the scattering. The denser the object is, the higher effective index is in this case. But if you do this on axis, so if you bring the light on angle, in general, it's going to be really hard to distinguish between the thickness or the concentration of those objects. So that's why that would be the challenge in 2D in general of the QPI. So that's why the holotomography is trying to solve the problem in 3D, because it means that somewhere here next to the nucleus you have a lipid droplet, but below it you have mitochondria. So if you do on axis, you get the maximal refractive index, or the sum of the refractive indices. You don't distinguish between the lipid droplet or the mitochondria below. So we're trying to give you the 3D arrangement of the organelles inside the cell by doing the 3D imaging. And this is just an example of the kind of images you're going to get out of it. So this is the reverse. So low refractive index is this. So it's white. The darker, the higher refractive index is. So you'll see the different compartments of the cells. You see the nucleus, nuclei, even the attachment of the cell to the glass. And if you need to look on the same with the same light path, with the same objective and everything else, using the same light source, on the left you have the bright field of the same optics. On the right you have, that's the difference in the resolution that we're talking about. Those 110 to 100 nanometers resolution is due to the computational reconstruction. So that's what the holotomography is. You computationally reconstruct the 3D information based on how the light is being scattered. And again, these really high resolution images. But why would you do holotomography at the end of the day? I mean, you saw so many other techniques out there. You probably use many of them already. So these are just some of the benefits of using holotomography. So in most cases, of course, it's going to be non-invasive. So you could image for months, same cell for months, assuming they have the nutrients in there to survive. Because it's the amount of light we use to image those cells is so little that you could image for months in this case. Also, it's quite fast. So it's always, the question is how fast is that? It's going to be faster than other techniques, it's going to be slower than other techniques. But we're talking about fast 3D, we're talking about seconds imaging a full volume in this case, or below a second in some cases. And the resolution, as you saw some of the images already, we're trying to push the resolution as much as we can to give you that insight where you can see the nanoparticle inside the cells and so on. And it's consistent. So that means it's not dependent on how long the fluorophore is inside the cell. Do we have any photo bleach? We're constantly imaging the same method across. So if something changes in the cell, you're going to see that change. And that was the example that I gave you at the beginning, where you see the cell literally being killed or being signaled to kill itself by the T cell in that case. And you see the changes without having to label anything in that case. So this is the step where I mentioned this so-called second generation holotomography. I'm not going to give you an intro on the first generation. Literally the presentation before it was about the first generation. We used to have the first generation microscope, and that's why I said the 2022 is the second generation holotomography. And I'll just give you a quick description. How do we get those images that you just saw? And I'll show you a few more in the second generation holotomography. So in our case, what we're doing here, if you want to go into the more details, this will be the paper to go and have a look at. So you see it's published in 2021. So what we're doing here, we're using an LED. So we're using low coherence. So we still need to have some sort of coherence for the measurement. Because at the end of the day, this microscope is a measuring tool that then takes the measurement, the refractive index, and creates the pretty pictures for the volume that you can visualize afterwards. But the way we do it, we're using something called digital micromirror device. So we're using a DMD with small mirrors where we can just change the angle of the light as we want. And then we do multiple patterns in a single slice, and then we do slices through. So that means the patterns that you see there, those are literally the patterns. We do four patterns in each slice. So we do slicing through, which means that we can go from 0 to 150 microns in the depth of the field of view. And then you get this kind of raw intensity images that gives you different information in each image, and then we reconstruct that with a computation reconstruction algorithm, which is a GPU-based reconstruction. And that's how you end up with this kind of images. So you see the contrast here, it was mentioned before, like whenever you use interferometer or use a laser as a light source, you're going to have some spectral noise. Now, because we're using an LED, you see the contrast there, you see literally the membrane attachment to the glass, all label free. And this is the kind of information that you can get. You can see all the things in here. Of course, in some cases, you're going to ask me, like, how do I know that that's a nucleus? I mean, morphologically, we can confirm that that's a nucleus. Of course, you can also label it once, confirm that that's really a nucleus, and now you know from here on. But I'll touch on that in a second. But you'll see the mitochondria, you see the whole cell in here. So you always have to remember it's label free, 3D, and quantitative, but it's not specific. So in some cases, whenever you look at the fluorescence imaging, there's always, you're at the mercy of labeling efficiency in some cases. So we'll look at this image and you'll assume that there's nothing or just barely anything in there, and then you realize there's a full cell next to the area of interest. So fluorescence is specific. You only see the thing that you tagged. So in this case, the mitotracker. In holotomography, you measure everything in the volume, regardless of what changes there. So if you flow in different medias and you flow in nanoparticles, you're going to see nanoparticles soon get bigger than the resolution. So these are just some features. I already mentioned the local coherence, and that gives us the really dense, or the really contrasting images in this case. I always want to emphasize on this. This is one of the biggest changes in holotomography or QPI in general. The fact that now this kind of images that you see here are all calibration free. So that means you don't have to go and adjust the lasers and make sure they have the perfect alignment and so on. In this case, we're using a single light path. So that means if I give the 10 minutes instruction to a high school kid, he can run the microscope. So basically, that's the whole point of it, because many of us would like to just understand the technique behind it, the physics and so on. But in many cases, you want to take your sample, the pressure sample, put it in the microscope, image it, get the data, and do the analysis and publish the paper. So you don't want to sit there for hours. And that's what we're trying to do here. We created this microscope now that you can literally put in a core facility where people don't have to be trained for hours every single week or every 2 months. You have to retrain them. We can train them once and they can run the samples themselves. So you don't need someone to do it for themselves. So we're trying to give as many options in terms of the labware. So we do work with standard dishes. So I know some of you are always concerned about the labware that you're going to use. We're trying to make it as open as possible. So also, because you want to image for a long time, you want to make sure that there's no Z drift. So that's why we're using a laser-based infrared autofocus to always scan for the glass. The glass is not going to move anywhere, but the cell might detach. So whenever you use a software autofocus, you're going to have that problem. So in our case, we've always been using this S22. Also, now I mentioned the 150 microns in Z. So that means now we can go to bigger samples. First generation, we couldn't do more than 40 microns due to the interferometer sensitivity and so on. Now we can go deeper because we're literally focusing as we go through the sample. And everything is incubated. So, of course, in this case, you can do long timers and so on. And you'll notice something here. So probably you're going to ask me, like, why do you do fluorescence imaging? Why would you want to do qualitative fluorescence imaging? So as I already mentioned, holotomography is great. You can image for months, but it's not specific. So whenever you want to confirm that there's something inside yourself is what you think it is, you can confirm it by using fluorescence. You don't have to image with the fluorescence. Fluorescence is just the add-on imaging method that is going to confirm that that really bright dot is indeed your lipid droplet or nanoparticles in some cases. So what we try to do here is make it as user-friendly as possible. So we also decouple the fluorescence from the holotomography. I just mentioned you could image every 10 seconds in holotomography, get the full 3D, label-free, without interfering with the cells in any way. But then maybe every hour or every 2 hours, you want to confirm that that object is where the thinking is. So that's why we decouple the fluorescence from the holotomography. And you can do up to 3 channels, 3D fluorescence. It's always good to mention that we've always been doing 3D fluorescence on top of the holotomography, because you have holotomography which is 3D. Why would you do 2D fluorescence on top of that? I already mentioned this. I'm not going to go into too much details. This is just a few examples of the types of labware that we can work with. As long as it is glass bottom of that thickness, of course, optics, so we're at the mercy of the objective that we work with. And it can be polymers, so BD treat and so on like that. So these are just some examples. Off the shelf, the only custom dish is the 96 well. And I mentioned already, if you bring the light and axis together 2D. So in our case, we bring the light in an angle. So that's why we cannot work with wells or dishes that have the wall bigger than the width, because physically we cannot project the light on the bottom. So physics, we cannot get around it. So optically transparent, of course, the sample has to be transparent because we want to pass the light through. The light scatters, we measure the scattering of the light, and we get those images. And keep in mind that we can work with different types of medium. So we can work with hydrogels, matrigels. I know some people do organoids. We can image them perfectly well, even agarose and stuff like that. Usually the thickness of the sample, I'll say 150, and that's because we're again at the mercy of that objective, which has a specific free working distance. It's inverted microscopes, so at some point you're going to hit the glass from the bottom. I'm not going to go into too much details, but basically, if any, the objects that we can image, I know some of you will be like, oh, what would be the ideal type of sample that you put on this microscope? I'll be like, why would you want to see just one sample? So in this case, because of the resolution of 150, we're talking about half a micron bacteria all the way to 150, 200 microns organoid. So that would be the range in our case. So you can image bacteria, you can image yeast, suspension cells, because we don't care if the cells are attached to the glass or not, because we don't move the sample when we apply. So in classical holotomography, or the first generation, or other QPR systems, if something moves in the field of view because of the 2 lasers, the measurement is going to be skewed. So in our case, we move the objective underneath instead of moving the sample. So the sample is only moving X and Y, and then when we click acquire, it's doing the Z-scan from bottom to the top with the objective without touching it. And that's why we can do T-cell assays, for example. So you can have an organoid with T-cells coming and attacking it as well. And this is just a few examples. You don't have to do it every 10 seconds. You don't have to image every hour. In this case, you can choose whatever the time you want. So this is just an example of primary adipocytes on the left, differentiated adipocytes, and heteroid cells. So you can see the interaction of the organelles inside, or you want to look at 72 hours in growth of the lipid droplets in this case. So you have full freedom to decide what kind of experiment you're going to do in this case. Of course, if you do every 20 seconds for 24 hours, you might end up with a few terabytes of data, but that's a different story. As I said, you can go crazy and image for days. I know this experiment, they've done it for 45 days. Of course, they don't image continuously 45 days, but you could, if you have a system that you don't mind keeping it imaging for 45 days, but you'll see the increase because these cells are not dividing anymore, they just differentiate, so they're going to grow. So you see the lipid droplets growing every single day, so you can track them. Because you put them into a multi-well plate, that means you can put them like this or like this. You literally can image the same position every single time, assuming the cells didn't move from the field of view. So we save the position X and Y, and that we use the autofocus to find the cluster. Which means in some cases you might image for 2 hours, take the dish out in the hood, change, add some treatment, put it back in, and continue imaging from there. Or you could do it literally inside using the microfluidic devices, which I'll show you in a second. This is another example of adipocytes, differentiation, and you can see the increased lipid droplets. So, as I said, no background, no calibration. So you literally take the sample from the incubator, put it into the incubator inside the microscope, and you can image them at 100% confluency, semi-adherent or suspension cells, and so on. And as I mentioned, the fluorescence. I already mentioned it, so I'm not going to go into more details, but we're doing 3D fluorescence on top of the holotomography to confirm that that's indeed mitochondria and so on. You can do that in monolayers. Some people call this 2D, but nothing is 2D ever in biology. And then you can do it in 3D systems like organoids, spheroids, and on C. elegans and whatever else. So this is a really cool example that I want to show you if you want to remember anything. So this is a microfluidic device. You can use microfluidic devices inside the microscope. As you image every 20 seconds, while you image, you flow reactive oxygen species, and you can see the effect of those reactive oxygen species onto the cells. So you see these ones are not going to recover, but then you bring the fresh medium in, and this one is going to start. You see those stress vacuoles, and they're going to go down one by one. So this is what I meant, that you could do 45 days by using flows, like microfluidic devices to exchange the medium, for example. So you could do that as well. Or you do it where you want to image before, during, and after the treatment, in this case. All while you're imaging without any issues. Of course, you will have to have a flow, a specific flow that is not too far up, but you're not disturbed by the flow. You see the flow going through in some positions, but because the cells are adherent in this case, they're fine. Or you can go massive tiles. You can do that as you wish. So this is a 4 by 4 millimeter. This is the same example I showed you before with the 3 adipocytes, where you look at the liquid droplets in this case. And everything is at the same resolution because it's a single objective. It's probably someone who might ask me later. Or you're looking at apoptosis. So in this case, if you follow the cells, you can see the events without any labels. But you see this condensation happening inside the cell before the bubbling, which you normally see in bright field. You don't see this in bright field. That's the big difference. So I know some people would assume that this is like a fancy bright field. It's not the bright field, because bright field is, in this case, we measure effectiveness index, so it's going to give you the dry mass. And because it's 3D, it's going to give you the concentration of the lipids or proteins in that case. So it's just another few examples of slow and fast time lapses where you can see the lipid droplets interacting with mitochondria and the things inside the cell. So as I said, if you can see something in 3D, like you see these white dots, you can segment them. If you can segment them, you can quantify them. So in this case, because it's 3D and it's quantitative, you get the volume of the yellow mask here. You can get, okay, this is 5 cubic microns, and it has 5 picograms of lipids. That means you can quantify the you get the concentration for each individual lipid droplet inside the cells. So that's the power of the quantitative 3D at this resolution. I'm not going into the specs, but that's probably some of you are going to ask me about the objectives and stuff. So this is the system that we're using. For X1, so we now have the plus and we have the mini. So I'll give you, if you come at the booth, we can talk a bit more about the different setups. I just want to briefly show you this, the big boy. So it doesn't look like a microscope, I know. It looks like a beer fridge or wine fridge, whatever you call it. It doesn't look like a microscope, I know. So this is the big one. We have the smaller one in here for this week, the live one. So if you have any samples, you want to try something, feel free to let us know. We are keen to try any real samples or any normal samples. It's up to you. So what we try to do here is to take the initial X1 that we launched in 2022 and make it better, basically. So we do now have different wavelengths as well. So whenever you image in holotomography, you can use whatever wavelength you want. There's a limit, of course, on the short wavelengths and longer wavelengths. But initially, we started with the blue light because blue light is giving you higher resolution. In the case of organoids, for example, the blue light is not as great. So that's why we introduced now the green light, the red light for the penetration, the bigger objects, and then the green light for the photosynthetic cells, because apparently chloroplast can absorb the blue and the red, but it's going to pass the green. We don't want absorption here. We want the refraction of the light as it passes the sample. And now we do have the bigger field of view. So think about this. You saw the 40x. We do get 309 microns field of view on a 40x objective. So that's the way we managed this is using a CXP camera and changing a few things inside the microscope. So that means now you can image massive area itself. And we have a preview camera, and we can also do a more sensitive fluorescence now with the CMOS course. So you have one camera for the fluorescence, one camera for the holotomography, overlay the two. So now you don't have to use too much intensity of the LEDs in the fluorescence and exposure time and so on. So, which means we can go down below a second if you do smaller volume in this camera, and means that bigger field of view, more cells and single field of view, better for analysis in this case. Or if you want to do higher throughput, now going from an X1 to a bus, now we can do the same area within a single acquisition, one field of view in one well. So that means in theory you can image a 96 well with this amount of volume every 26 minutes for as long as you like. And of course, bigger objects, now you can fit them in a single field of view without any tiles, and the information you get there is quite nice. That's just a quick difference between the blue light versus the red light due to the penetration. Of course, the blue one is going to give you the highest resolution, but this one is going to give you the more information in there. And this is just an example of organoid growing imaging every half an hour for, I think it was 60 hours. So this is the maximum projections. It's not intensity, it's refractive index in this case, but we just call it mid. And this is a single slice that you look at out of those 150 slices. And if you need to look at it in 3D, it will look like this. So this is just to rotate. You can rotate and visualize it, use the graphics card of the computer to visualize it in this case. And just this is an example of the blue versus green in plants. As you can see, it's really shady, like you have like just blurry information here. When you switch to the green light, now you don't have the absorption of the chloroplast in that case. And this is some other time lapses. You can time lapse on plants as well. So I know you guys have different samples, so that's why I saw some bacteria, some cancer cells, and so on. So I just wanted to show you different things. And because we have those three colors, we have the classical bright field, color bright field as well. So you can do holotomography, channel 1, channel 2, channel 3, 3D fluorescence plus bright field in every single position in every single time point if you want to. Of course, you don't have to, but in reality, you're going to use the holotomography and bright field most of the cases. Bright field is not going to give you as much information, but it's always nice to see the complementary of other stuff. So I know we're looking for the coffee and we're running late. So just to give you a quick overview, we do have different flavors of fluorescence. I know it sounds weird, but I know some people want the really sensitive camera for the fluorescence. Some people want the spin disc, and now we do have the spin disc for focal attachment on the holotomography as well. Which means that we're using a classical fluorescence module, which is the FLX that we created, which gives you really sensitive fluorescence, and then you get this kind of images. So holotomography, different channels overlaid with really sensitive fluorescence, so you don't have to bombard your cells with a lot of light to get these pretty pictures. And of course, you can overlay them all together, and you can do that in bigger objects. So you can do live, of course, but you can also fix. Some people want to do fix because we already fix it on other systems. And the maximum flexibility, whatever you want to call it, would be the spin disc, because the spin disc is the spin disc for focal, but you can also use this wide field in this case. And this is just an example of the spinning disc on an organoid, in this case like that. So different channels, in the case of the spin disc, you can go up to 8 channels potentially. So this is just a summary. I know we're running late, so I'll try to leave it as it is. And these are just some key applications. If you have any specific applications that might not be here already, so these are the things that we've already tried multiple times. I know some of the things might be yours in here. And also, this is just a quick overview of the systems that we have now, going from the really compact, which you'll see literally in the room over there, and then we have the OG, so we're the one we started with, which is the X1 back in 2022, and in 24, 25, we launched these guys one after the other. So initially we had this, and now we just have the spin disc as well as an option. So higher throughput, more sensitive fluorescence, more compact, so single lab use. Potentially these guys are going to be in the facility or multi-user, multi-lab setup, and that's how we kind of split them in a way, because we worked from this way upwards, and then we launched the mini because there was a need for a compact system that you could put in a lab, so IVF and other places they might want to do research as well. So that was me. Please feel free to ask questions. Please feel free to run to the coffee and ask me afterwards. Also, I just noticed that there were a few talks about the biofilms, bacteria and stuff. This is literally hot out of the oven. They created a biofilm app node because we're going to go to the Micro 2026, the ISM in Washington. So I just literally overnight they launched this app node about the biofilms. We have other app nodes for other things, so feel free to ask me. As I said, feel free to bring samples if you have any.
very much. I have a question. You mentioned that it's feasible alive and fix the cells or tissues. Do you see a difference in the acquisition and so the resolution that you get out of it at the end once the tissue is fixed?
It's not the resolution, it's the difference in the material that you have left. So by fixation, so you have fixation and then potentially polymerization and so on, you lose mass. So it's literally you remove lipids and so on. So I would never compare live holotomography with a fixed holotomography, just because of that reason. We see like we've done image time lapse, and then at the last minute we've done one image, fix them, image them again, fix them, stain them, image them again. You see a drop in the dry mass itself because you remove material from there.
And then I have another question. You mentioned you did it on C. elegans or something?
Yeah.
Okay.
So we've done C. elegans. I don't have any examples at the moment, but yeah, we've done literally last week in Switzerland. We've done C. elegans as well. So they do a lot of C. elegans over there.
You said you offers high resolution, even to solve distance like 100 nanometers, thanks to scientific approach. Could you please explain what it means?
Yeah. So in this case, we use probably it's hard to see, but it's a what we're using is a big condenser because we still need a high numeric aperture to project the light in an angle. So we're using different parts of the numeric aperture. So in theory, we can project light as we want from the DMD. So think about it more like the easiest way to describe it for myself is like the light comes from here in an angle, hits the numeric aperture, and then goes onto the sample. But it's a full illumination in that case. So that's why if you want to go more in details, I'll check the paper because it's called Pepsi ODT. I don't know what they want for this name. So it's purely efficient structure illumination, optical diffraction tomography. It's a mouthful. Okay. So if you want to go in more details, I'll give it a try.
You have shown many images mentioning that contrast comes from the index of refraction, but I haven't seen a map superposed on the image with the index of refraction.
It's the
Like you have
one
the
of the first ones.
across.
So it's one of the first ones when I said that the white is the low refractive index, and let me show you quickly.
For instance, in any of these, can you put them up?
Yeah. It's just because they didn't put it in this case. They put just the what's it called? Actually, the example that I was thinking about is the first one that I showed you, which is this one. So this one says like this. It's not in every image, but you can put it in every image from the software. You can input even the model of the system that you use and so on. So this is usually the way we show the images. This is inverted, so the high refractive index is black in this case.
Okay, so in this image, what you practically does the color map, yeah?
Yeah.
And how do you get rid of the whatever comes from the intensity? You normalize and eliminate intensity. There's no contribution of intensity when I do a distinction between this pixel and this pixel.
No, it's uniform across the whole 3D. So in this case here, you're looking at the maximum projection, of course, of all the stuff. But if you go slice by slice, you get this kind of information. So in each position, so think about this is the object. In this position, you have 1.35. In this position, you have 1.36. And here you see the highest refractive index from all the slices. But when you go slice by slice through them, you get individual ones. And that's down to the resolution Z as well, of course. So that's this one's 156 nanometers resolution.
I know that for some micro tissues or organoids, you need tissue clearing in order to see better and with more resolution for fluorescent signals. Is it still necessary for this type of visualization to?
It goes back to the question about the fixation. So if you remove things from there, you're going to lose contrast in this case. Of course, the bigger the object, the denser the object, the more scatter is going to be. We want the scatter because that's the measurement itself. Too much scatter is going to be hard to track back where the signal was supposed to be. So as long as the tissue is not too thick, the thicker the tissue is, the more scattering you're going to have. So by clearing, you do that. In our case, that's why I say 100, 150 microns. That should be ideal, even for organoids. So the example that I showed you, the organoids, they were like 100 microns.
And the second short one, how much time does it take for, let's say, time-lapse microscopy overnight file to be reconstructed? So for the image analysis.
Depends. You mean after acquisition?
after acquisition, how long does it take to generate
The 3D.
visual,
So you mean the processing
the
itself.
3D.
So that would be the
Yes,
deconvolution. Let's call it deconvolution.
Yeah, the convolution.
Um, So acquisition takes, if you do time lapse, you could image every 6 seconds once in the position. Processing to see this image takes about 30 to 40 seconds depending on the Z itself. So if you image every 7 seconds, you're going to have a backlog of the processing because it's computation construction. You need to give all those 4 images times 150, so 560, or whatever the quick math is going to be. To reconstruct that is going to go into the GPU of the computer to give you this information. So that's what, 6 to 30 to 40, so 5 times more time taken to reconstruct the data.
I didn't quite get the question, but I think I have a similar question regarding the refractive index determination, because you said you were using it quantitatively to determine concentrations and so on. Do you have a clever trick there, since you're mainly measuring refractive index differences and objects are fuzzy and small and so on? It's not super easy to get the absolute refractive index, right?
It's not an absolute refractive index. So that's always in QPI. You never get absolute refractive index. You get a reference to the medium itself.
Yeah.
So you always have to give the algorithm something. So you need to know the refractive index of the medium itself.
But you only need it from outside, and then you can use the inside the cell as
In
well.
this case,
The
yes.
cell is not the same medium as well as.
It doesn't matter in this case.
Really?
Because what we do here, we don't calibrate based on the medium as you do in the first generation holotomography or the classical QPI. We measure everything in the volume as it is. So that means if the light scatters a lot from here, that means this object here is really dense when the next slice is below. So that's what we do Z step through.
Yeah.
We do overlapping in Z itself as well. And each pattern apparently is supposed to have some specific information of the light being scattered. That's the way I would describe it. So I didn't go into the physics of it too much myself.
Okay, if there are no other questions, let us thank the speaker again.